Continuing with the chapter number 5, biological molecules, we have talked about three basic biological molecules: carbohydrates, proteins, lipids, and we also talked about the tests. Food tests के अंदर हमने कौन-कौन से cover किए थे? Iodine test for starch, genetic test for हमने कौन सा किया था? Glucose. उसके बाद हमने Burette test किया था for proteins, then the fat emulsion test for lipids, और ये हमने सारे tests कल cover किए थे. Today we'll be starting off with enzymes. The third topic of your biological model. Enzymes क्या है? By the name, they are actually proteins. ये क्या है? Enzymes are proteins. Now they are biological catalysts. Biological because they are made in living cells. हम इनको catalyst क्या होता है? If we talk in chemistry terms, catalyst is anything that can speed up the chemical reaction. Catalyst क्या होता है? Anything that can speed up the chemical reaction. But biological catalyst क्या है? They are biological because they are made in living cells. And they are called catalysts because they speed up the rate of chemical reactions. They are specific to one particular substrate, as the active site of the enzyme where the substrate attaches is a complementary shape to the substrate, known as lock and key hypothesis. For example, so catalysts हैं because they speed up the chemical reaction, then they attach to the substrate. अब दो चीजें हमने यहाँ पे कुछ terms आएंगी. The first term you should know about enzymes क्या है? Biological catalyst. The first term. सेकेंड आपने याद रखना है टर्म कैटलिस्ट का क्या मतलब होता है? एनीथिंग दैट स्पीड्स अप द केमिकल रिएक्शन. नंबर 3, आपको एक चीज पता नहीं, सबस्ट्रेट. सबस्ट्रेट इज द मॉलिक्यूल ऑन व्हिच द एंजाइम एक्ट्स. फॉर एग्जांपल, दिस इज स्टार्च. ये सबस्ट्रेट है मेरा, दिस इज सबस्ट्रेट, एंड द नेम ऑफ द मॉलिक्यूल सबस्ट्रेट, जो भी मेरा सबस्ट्रेट है जिसके ऊपर अभी एंजाइम एक्ट करेगा, उसका नाम है स्टार्च. ठीक हो गया? नाउ साथ ही मेरे पास एक एंजाइम है. एंजाइम इज amylase. ठीक है? Now I know that this enzyme, which is the amylase, is complementary to the shape of this substrate. Complementary का क्या मतलब होता है? Exactly the same, which can join. फिर आपको पता होना चाहिए what is an active site. अब लॉक एंड की हाइपोथेसिस क्या होती है? मैं कहता हूँ this is the enzyme and it has to attach itself to this substrate because only complementary shape can attach. अगर मैं आपको कहता हूँ ये मेरा एक substrate है and I have two types of enzyme, one of them is this and one of them is this is my substrate and this is enzyme 1 and this is enzyme 2. Now which one of these by shape that you can tell can attach itself to this? Can E2 attach to the substrate or can E1 attach? Obviously E1 has a shape complementary to the substrate. Only E1 enzyme 1 can act with the substrate and then the whole chemical reaction would be speeded up. क्योंकि क्या होता है, starch जब break down हो रहा है, now starch takes a lot of time to break down. तो उसको तेज करने के लिए, how to speed up the breakdown of starch, we use an enzyme and that enzyme is known as amylase enzyme को हम use करते हैं. Now this whole process is known as the lock and key hypothesis. इसके अंदर आपको जो substrate है that is the key and enzyme is the lock. तो substrate अगर ये आपके पास for example enzyme है, this is the enzyme, this would be the substrate and this would attach, ये आपके पास substrate है and this is the enzyme. This would attach as a key in that lock and that lock is the enzyme. Other than this, you should know that these enzymes are affected by changes in temperature and pH. Temperature and pH do affect the activity of these enzymes, and also enzymes are not used up in the reaction they catalyze. But obviously, you know that reactants are converted into products. Starch जो है, starch would be converted into glucose molecules, लेकिन enzyme would not be changed. Enzyme would remain as it is, as it was in the start of the reaction as well. So this is the basics of enzymes, active sites, substrate, catalyst, and lock and key hypothesis.